Welcome to our video exploring the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Omastar and its connection to Cloyster. Ready? Let's go! The mollusks, mollusca, from the Latin molluscus meaning soft, are a highly diverse and species-rich phylum of tissue animals, Eumetazoa. They live predominantly in the sea, marine, but some forms also occur on land and in freshwater. The branch of zoology that deals specifically with mollusks is called malacology, from ancient Greek malakos, soft, or malacozoology. The body of mollusks is originally unsegmented, strictly bilaterally symmetrical, and divided into 4 regions. The head at the anterior end of the body, equipped with a rasping tongue, radula, and more or less distinctly separated, the muscular foot located ventrally, on the belly side, the visceral mass located dorsally, on the back side, more or less set apart as a visceral sac, and the mantle, pallium, which covers or encloses the visceral mass dorsally. Head and foot, which together form a functional unit responsible for locomotion, food intake, and, closely linked to these, information processing, are collectively referred to as the cephalopodium. The mantle and visceral mass, which as a functional unit primarily carry out the fundamental life functions, are therefore grouped together as the visceropallium. The foot in most mollusk groups is developed as an organ for creeping, burrowing, or attachment, with the creeping attachment function considered the original one. The outer cell layer of the mantle epithelium, secretes in many species an external skeleton of calcium carbonate to better protect the visceral mass. This external skeleton appears in various forms. It may consist of an episphere, i.e. a chitinous layer, cuticle, into which numerous small carious needles, spicules, are embedded. It may form a kind of plate-like armor, or it may develop into an elongated or spirally coiled shell into which the animal can fully withdraw. Plate-like or shell-like external skeletons are widespread among mollusks and are also referred to as shells. Calcareous spicules are considered a primitive feature and are found only in worm mollusks, Aplacophora, and chitons, Polyplacophora, the latter also forming plate-like structures. The epithelium of the cephalopodium often bears cilia and numerous glands that secrete various substances. The mantle forms, originally at the posterior end of the body, a more or less voluminous invagination, the mantle cavity. Within it lie organs known as ctenidia, comb organs. Together with many small cilia located in the mantle epithelium, they generate a water current that ensures continuous water exchange between the mantle cavity and the surrounding environment, since the intestine and the excretory ducts, see below, open into the mantle cavity. The mantle epithelium of the mantle cavity also serves for oxygen uptake and carbon dioxide release. In larger mollusks, with the exception of land snails and monoplacophorans, the ctenidia also function as gills. The excretory system of mollusks is considered unique. Ultrafiltration of the primary urine from the hemolymph, a primitive type of blood, occurs through the walls of the atria, auricles of the heart, into the pericardium, the coelomic cavity surrounding the heart. The pericardial epithelium, lined with podocytes and lying adjacent to the atria, is historically referred to as pericardial glands. Filtration is enabled by the blood pressure generated by the heart muscle. The primary urine, pericardial fluid, then passes through the pericardioducts, also called nephridia, metanephridia, or nephridial canals, one per body half, ciliated ducts that connect the pericardium at one end with the mantle cavity at the other. In higher mollusks, the pericardioducts are relatively complex in sections, and these sections are then referred to as kidneys. Within the pericardioducts, water and vital substances contained in the primary urine are reabsorbed, producing the secondary urine, which is discharged into the mantle cavity. The molluscan excretory system thus resembles that of vertebrates in its structure. The pericardial glands and pericardium function analogously to the glomeruli and Bowman's capsules of vertebrate kidneys, while the pericardioducts correspond to the renal tubules. While some groups of higher mollusks, Eumollusca, Testaria, exhibit a relatively conservative body plan close to the presumed ancestral design, with creeping foot, radula, simple external skeleton serving purely protective functions, strict bilateral symmetry, and anus at the posterior end, e.g. monoplacophorans, chitons, the latter having developed pseudosegmentation through multiplication of C. tenidia and a multiplated shell. Other groups show much stronger modifications. In cephalopods, cephalopoda, the original body axes are rotated by 90 degrees, so that the original dorsal side forms the posterior end, while the original posterior end with anus and mantle cavity lies ventrally near the head. Accordingly, the cephalopodium forms the anterior end, bearing a crown of tentacles or arms arranged around the mouth opening. The shell not only provides protection, but also functions as part of a hydrostatic apparatus, regulating buoyancy. In modern cephalopods, coleoidea, the buoyancy organ has been internalized or greatly reduced. In bivalves, bivalvia, the head and radula are reduced, the foot is transformed into a burrowing organ, and the shell is developed as a two-valved structure. In gastropods, snails, bilateral symmetry is partially reduced, accompanied by a 180 degrees torsion of the visceral mass. As a result, in snails the mantle cavity and anus are oriented toward the head. The digestive system of mollusks varies considerably according to their physiological needs. Particularly stable biopolymers are often found in the feeding and grinding structures, radula or parrot beak-like jaws of cephalopods in the form of chitin. Some herbivorous mollusks also employ endogenous cellulases in their digestive systems down cellulose, as demonstrated in certain snails such as the Roman snail Helix pomatia and in some bivalves, Corbicula japonica and Lyrodus pediculatus. The origin of the cellulase gene is traced back to the last common ancestor of the Bilateria. Sensory cells occur in the form of tactile, olfactory, gustatory, and photoreceptor cells. While simple photoreceptor cells are present in most groups, cephalopods, gastropods, and some bivalves have developed true eyes. The sensory organs are particularly innervated by the cerebral ganglion. Their highest level of organization is reached in the eyes of cephalopods, which, in their performance and structure, are comparable to the eyes of vertebrates. They may, however, develop differently. Other sensory organs are the frequently present tentacles, which serve partly for mechanoreception, sense of touch, chemoreception, taste, olfaction, smell, photoreception, light perception, as well as the perception of water currents. Another characteristic sensory organ is the osphradium, which in aquatic forms lies at the edge of the mantle, protrudes into the mantle cavity, and serves chemical sensory perception. Snails, gastropoda. Snails originally move by creeping. Typical land snails pull the posterior end of their foot forward at regular small intervals and set it down again, producing wave-like lines on the underside of the foot. Bivalves, bivalvia. Typical bivalves change their position only slowly. They seek a suitable place to burrow into or attach themselves with their muscular foot. By contracting the foot muscles, they can alter their position. In some bivalves, e.g. mussels such as Mytilus or Dreissena, additional byssus threads are secreted from the foot groove, allowing them to attach to solid substrates or other muscles and thereby change their position. Cephalopods, cephalopoda. All cephalopods can draw respiratory water into the mantle cavity and then expel it under pressure through the funnel. This produces a jet propulsion effect and enables relatively high speeds, further supported in many squids by opening and rapidly closing the arm crown equipped with swimming membranes. Especially the decapod squids possess fins or a fin fringe on the outer mantle, which they use to generate propulsion through wave-like movements. This allows slower, more controlled locomotion. Nautiluses, with their mantle largely hidden within the shell, lack fins entirely. Silk and shell production. Many mollusks produce silk or silk proteins. Bivalves, such as mussels, produce byssus to attach themselves, often only during the juvenile stage. Byssus is homologous in structure to insect silk, containing the silk proteins fibroin and sericin. Many representatives of various shelled mollusk classes, including some cephalopods, such as nautiluses, form shells whose innermost layer, the hypostracum, consists essentially of mother-of-pearl, nacre, a biological composite of calcium carbonate and fibroin-like proteins. The genetic basis for silk protein production is traced back to the common ancestor of mollusks and arthropods. Over the course of evolution, this gave rise to diverse uses, secretion organs, and molecular structures of the respective silks, many of which are not yet fully understood. Distribution and fossil record. Mollusks inhabit all environments on Earth except the polar regions and high mountains. Most mollusks live in the sea, with almost all groups being exclusively marine: cephalopods, monoplacophorans, aplacophorans, chitons. Bivalves also occur in freshwater and sometimes even in moist soil, such as pea clams, Pisidium. All other habitats are colonized by the largest mollusk group, the snails. The total number of species is given very differently, ranging from well over 100,000 species to sometimes only 50 to 60,000. The main reasons for these discrepancies are unclear species delimitations and, above all, the lack of a comprehensive overview of the diversity of the various subgroups. Because of their well-preserved external skeletons of calcium carbonate, mollusks have a generally dense fossil record. The oldest fossils that can be confidently attributed to mollusks are found in the small shelly fauna of the earliest Cambrian. However, the phylum most likely originated already in the late Precambrian. The organism Kimberella, known from the Ediacara fauna, is considered a possible stem group representative of the mollusks. There is still uncertainty about the common basic body plan of the mollusk groups living today, the mollusk crown group. Some researchers assume that the most recent common ancestor of the crown group was a small, worm-like species barely half a centimeter long, with an elongated digestive tract, anus at the posterior end of the body, and relatively strict bilateral symmetry. This ancestor is thought to have lacked a closed shell, instead possessing an episphere and a foot with a ciliated gliding sole, similar to some modern worm mollusks. Others, based on the fossil record and observations of extant species, argue that worm mollusks are not the most primitive forms, but rather derived from chitin-like ancestors. This calls into question the reconstruction of the ancestral mollusk as a worm mollusk-like organism. The evolution of mollusk classes and their phylogenetic relationships to one another is not yet fully resolved. Together with other animal phyla, mollusks are placed within the larger group Lophotrichozoa. Within this group, the Entoprocta, also known as Camptozoa, are currently considered the closest relatives. The earlier, often expressed assumption of a direct relationship between mollusks and annelids, segmented worms, is now rarely discussed. As a central and systematically significant characteristic of the mollusk phylum, in the sense of an autapomorphy, a shared derived trait, the radula, rasping tongue, is considered defining. Other central morphological features of the original mollusk body plan include the tripartite division of the body into head, foot, and visceral mass, the development of a mantle with a mantle groove, the reduction of the column to the pericardial system, and the formation of an originally always open circulatory system with a ventricle and one or more arteries. In addition, the shell is a significant feature of almost all mollusks, although the primary single-piece shell with its typical three-layered structure occurs only in the shelled mollusks, Conchifera. Clymeniida, Clymene, are an order of late Paleozoic ammonites, Ammonoidea. They serve as important index fossils of the Devonian. Their shells vary from thin and discoidal to thick. The umbilicus ranges from wide to closed. Unlike all other ammonoids, the siphuncle runs dorsally. The lobe formula is Li, with many modifications during the group's evolution, such as the development of E, A, and U lobes. The forms are generally weakly ornamented, with growth lines or faint ribs. The Clymenidae are a family within the order Clymenida, a group of late Paleozoic ammonoids. They lived during the Upper Devonian, ca. 370 to 360 million years ago, and are important index fossils of that time. Their name derives from the type genus Clymenia. The Clymenidae were marine cephalopods that probably lived in the open ocean. Their shell morphology suggests different swimming and lifestyles, from free swimming to near-bottom dwelling. The dorsal siphuncle may have been a special adaptation for buoyancy or shell stability. The order Clymenida was first described by Alpheus Hyatt in 1884. Fossils from the German Devonian, e.g. Arnsberg, Beckham, are among the most famous finds. That's it for now. In the next episode, we'll take a closer look at Kabutops and its surprising connection to all arthropod Pokemon. Don't forget to subscribe to our channel, leave a like, and check in next time.